just, oh, I just randomly put stuff up there, but I have nothing to back it. Okay, so you need to make sure, if you don't have it written on that, that you're making sure that you're sharing that information when you guys come up. Hey Siri, she's talking to me again. What did I say?
Hey Siri.
No, not now. She had already talked to me before I said that. And there's words that I say that picks her up. But anyway, when we get done doing that, then we said we collectively have to answer that last slide. How is tissue culturing going to be, I don't know, helping to feed the 9 billion mouths that are projected to be on this planet? So we will, after we go through all of this, we're going to kind of have that conversation, and we're going to add that last
So
bit
add
to
the
it.
slide after.
Huh?
We were supposed to add the slide after it.
That would make sense.
So yeah.
Yeah.
We were supposed to do it before.
The slide after, what do you mean?
What are you talking about?
Okay, I'm confused. The very, very last slide, it asks the question, how is tissue culturing going to be able to combat world hunger or feed the world or whatever? After everybody gets
That's
done
how
doing the
it
presentation, we're collectively going to do that.
That's how it is.
That's selective hearing again, isn't it?
Maybe.
I just have poor hearing in general.
I think it's attention. I mean, I'm not trying to be a butt, but I really think a lot of it is attention. You guys get, oh, I'm on my computer, or I'm on my Snapchat, or I'm on my whatever, and I think I'm listening, but I'm not really. I think there's a lot of it. I don't know that it's not hearing. You hear, I just don't think you listen, or you have the attention to it. Anyway, so we get done with doing that. I think everybody but Shelby, I didn't happen to see a link that you had added on that resource page. Maybe you did, I didn't happen to catch it. If you didn't, you need to add that there. When we get done, we are going to go ahead and get set up for our BT lab. The European corn borer larvae came in yesterday, so I've got them incubating right now, but we're going to get things set up to go ahead and be able to do that. So that's kind of just our plan for today.
So
Again,
is it just you put it in a petri dish, there's nothing done,
no agar or agar, whatever you want to call it. I just
gracious.
have a paper towel in there, and then you wet it, and then you're going to put that leaf in there, and then you're going to put the European corn borer eggs on top, and then they hatch and they go to town.
Oh my goodness, there's nothing too complicated.
It isn't. Most of the stuff that we do, guys, is not really complicated. It's just, do we follow directions? Somebody grab the door for Dawson if you would, please. I was going to say something after that.
Wrong
direction.
Yeah, I know.
You want to warm?
Keys.
Okay. Thank you.
Anyway, if we are continuing on, again, investigating or looking at different selected breeding processes, propagation processes. Again, when we think about plant biotech, lots of things. You guys have seen there's lots of things that we can do. Are there varying opinions about these things? About the legitimacy of them, about how effective they are there other alternatives, whatever. Again, I'm not trying to tell you that this is going to be the cure-all for everything when it comes to combating world hunger. Is it one item? Okay, is it one item that we can use that is going to help us do that? And then again, once we've used this biotech, we've engineered, we've edited, we've done whatever to our crop, now we're talking about how are we going to propagate this. And this is, again, one method. So I know Shelby's been in my horticulture class, and you told us yesterday, is there just one way to propagate a plant?
Gosh,
No. Gosh, no. Okay, there's lots of different ways, and this just happens to be one way that is very commonly used in biotech. So, plant tissue culture, micropropagation are the same thing. Okay, means the same thing, so that's why I've got both of them up there. And I do believe, Connor, you are the first one up. So if you want to come up and you want to go through your slide, and then I do have the video link open for you, so you'll just have to escape out, and then you'll have to just open up your video and then go ahead and go full screen.
I think we're going to start with the video.
You're going to start with the video? Okay.
What's your section?
He is
Define.
defining what plant tissue culture is or micropropagation.
Definition.
And I don't know if you'll have to turn that up or not.
Natural vegetation, propagation, and plants. In 1902, G. Haberland noticed that the plant cells can be grown in synthetic medium.
Push pause.
The discovery.
What is a synthetic medium?
It's like the
not
it's like the
agar, ain't it?
There you go, it's the agar. So when we think
Agar.
about typically when we are propagating plants, where do we propagate them? In soil. So this is an alternative method that we are, it's artificial. Okay, it is going to be propagated in some other media other than soil. But again, is there an advantage to this? There's actually multiples, which Katie is going to allude to here in a little bit.
Thanks,
Go
Connie.
ahead.
By Haberland, that the plant cells have the capacity to grow in a nutrient medium in presence of sufficient light made an impact in plant propagation and crop improvement.
So this is like, so they said like since like they've been able to like do this in like the artificial light and in the agar, it's actually able to grow better than in the soil.
There's probably more nutrients if you use
I was gonna say, is there a lot more nutrients? I think there's a bigger issue coming yet. And actually, I keep saying that it's just one thing. No, there's like 10 things that make this amazing. Now, are there some things that make this really,
Of course.
yeah.
Well, like, because they're all the same, it's like they're trying to say plant over and over. There's that one thing wrong in my part of this piece.
There you go. And we'll kind of get into that as we go through. I'm gonna let you go.
The ability of a plant cell to give rise to a whole plant is called totipotency. The
Totipotency.
cell and tissue culture techniques have taken the method of asexual reproduction in plants. In this method, instead of taking cuttings from a plant, a cell or a few cells or a tissue is taken from a plant and cultured in suitable containers in which nutrient medium is present.
Pause.
The portion of
Okay, so what is unique about this? So normally, and again, I keep picking on you, Shelley, because you were in horticulture class, but what were some of the ways that we propagated?
Cutting seeds.
Cuttings and seeds. Thank you, Jay, not a horticulture person. But cuttings and seeds. So we had to have a mature plant. We had to take either the reproductive part, which would have been that seed, which is a result of sexual reproduction, or we had to take a stem cutting, or a root cutting, or a leaf cutting. Do we need an entire cutting? Do we need an entire seed?
They've seen a few cells.
There you go. We just need a few cells, and you think about the significance of that
You can
alone.
take a lot of plants from one plant.
There you go. We need one little section of tissue, and can we make millions upon millions of copies of that tissue? And I don't know if you caught before that it says every single plant cell has every single thing necessary to reproduce an entirely new plant. So it's not like us, like if we cut off our arm, it's not like we can plant our arm in the soil and it's all of a sudden it's gonna grow what we're missing. In plant world, can we do that?
Yes.
Yeah, we can, and that's what makes it so cool. And then again, you guys are gonna be telling me, why is this technology such a big deal? Why is that such a beneficial thing to be able to take such a small, teeny tiny amount of tissue and make millions of it? Okay, Connor, you're back up.
the plant that is taken as the desired plant is referred as explant. By this method, thousands of plants can be produced from a single desirable parent in one generation. These techniques require maintenance of sterile conditions in the medium.
Plus, press pause.
The
What did it say? It requires who?
I
Or
can't
what?
understand what that gal said.
I couldn't.
techniques require maintenance
A
of
maintenance.
Which one?
sterile conditions in the medium.
Sterile conditions.
What is sterile conditions?
I guess I got also
Your sterile conditions.
There
is
you
your-
go.
is your-
Sterile conditions. And again, guys, going back, connecting to what we've already done. When we are pouring dishes with nutrient agar in them, do I let you just sit and leave those dishes open to the air?
No.
Why not, Katie?
Due to bacteria.
Thank you, Katie. Katie, why not?
Yeah, I know,
What?
it's shocking.
if people would answer
What?
I said that I did not have a twin.
Oh. Anyway, why don't I let you leave that lid open?
Because bacteria.
There you go. There's bacteria that's floating in the air, and then that bacteria sits on that, and then all of a sudden, do we have contamination?
You have a lot of problems.
Then we got lots of problems. So when we think about this, okay, it's gotta be sterile conditions, whatever kind of language we got going on, which is sterile conditions. Go ahead, sir.
Eating. The plant needs to be sterilized before it is introduced into the medium. The medium is kept free from bacteria and fungi. Cells or tissues are grown in culture under a set-like condition. In the tissue culture, the explant often divides to give rise to an unorganized mass of tissue called callus. The callus or the explant is such will undergo differentiation into roots, shoots, or embryo-like structures is called embryos. This differentiation is dependent on the concentration and combination of the plant's growth substances like auxins, cyanins, gibberellins, etc.
Push pause. What are auxins, cytokinins, and gibberellins?
Like type of auger.
Nope. It's in the auger.
Yeah, that's not like all
Okay,
the
Shelby,
things
I'm
that
gonna go back and pick on you again. We talked about this in horticulture.
I said it's the things in the auger.
Yeah, but what is it specifically? What is it? Is it a nutrient in the auger? Is it a mineral in the auger? Is it a hormone in the auger? What is it?
The hormone.
Right there.
Ooh, lucky guess.
It's a hormone. Okay, and those hormones, again, going back to horticulture class, and I understand that not all of you guys have been in there. Those
Take your shot,
hormones, boys, are what causes these plants to divide cells, to multiply, to create different types of tissues. So they're loaded. So like I said, it's this teeny little wad of cells, and then we've got nutrients on there, we've got minerals on there, we've got hormones, and it just starts growing this massive that is called a callus. And it's really kind of weird looking when you actually get this to grow. It doesn't even look like a plant really. That's so that little picture of that little plant, not what it first looks like. It's just a wad of crap. Okay, that's green and it's growing. And then you cut it up, and then we're going to put it in other hormones that are going to cause it to differentiate out into other things. She keeps putting up there, or they keep typing up there, explain, explant. It's not explained, it's explant. That's what that new plant is an explant, not an explained.
Well,
She's
it's like-
saying an explant, but they're typing explain.
Well, it's like it's YouTube has like their own closed caption.
Right, just like the auxins and the cyaninin, that's cytokinins, which is not spelled anywhere close to what it really is.
Well, I don't think the accent helps much either.
No, correct. Go ahead.
In the medium, plants raised through tissue cultures are routinely used in agriculture, horticulture, and forestry. We'll use a diploid explant.
There, okay,
We
you
get
explain
a
this.
diploid plant like in any other vegetative propagation methods. Similarly, if we want to develop a haploid plant, it is possible to obtain it through tissue culture.
Press pause.
Develop
What's the difference between haploid and diploid?
Haploid is one.
Sex cells.
And
Diploid is two.
Woohoo! Go Connor.
I don't know. Because
method of haploid plants through tissue culture was discovered by Indian scientists Shipra Guha and Satish Maheshwari.
didn't know that, did ya?
I didn't.
Toyota?
With the
They're just named.
Toyota
Okay.
Christmas sales event is here.
So, if you go back, so what did you say is exactly the definition of this? It
It's
is,
taking a very small piece of tissue from a plant and able to make a whole new one.
okay, would you guys agree with that based on what the video you just saw?
I sure will.
Yeah.
Well, you could probably put it like in a sterile environment or
Oh.
like.
Keep
Oh,
going, keep going.
it's in here, Jay.
I'm just saying.
Okay, hostile people.
That was a good movie.
Go ahead.
Okay, so just like to kind of recap, like they're able to grow cells in laboratory using HEPA filtered air, so it's just like sterile.
That's that big goofy looking thing that we have right out here that sits on my table. That's a laminar airflow hood that has a HEPA filter
That's
in it.
that's nothing.
That you do all of, when we do
Oh,
our
yeah,
tissue
yeah.
culturing, that you do it all in this like area, and that air that's going in there is completely and totally filtered.
Huh.
So it keeps the bacteria out of your nutrient media.
Like and like what they talk about in the video, like with the hormones on it, they put it in agar,
Mhm.
and this is in sterile containers, so there's no spread of disease.
Okay, so is this like done in a greenhouse setting? Or is this
No.
done in a laboratory?
Laboratory.
This is done in a laboratory. Now, eventually, are these plants gonna end up in a greenhouse or outside?
Yeah.
Absolutely.
Yeah.
But to get them started, we have to do this in very sterile setting and everything's gonna be done in a lab and blah, okay? Thank you.
I did good.
After a little more encouragement from Murphy Puppy this morning.
No, I had complete intention of doing today during my first hour.
Okay.
Yeah, in your other room.
Oh,
Yeah.
well.
We're supposed to be drawing pictures of something, Connor.
Okay, so
Yeah?
Katie is going to share with us the major advantages of doing said tissue culturing and or micropropagation. So,
better be a video.
there is not.
You don't need a video.
You don't need to. Go ahead, Katie.
Okay, so some of them are there's rapid multiplication in a short time in a small space, like it explained in this picture here. They take the little plant and see that in a small, really short.
And one of the things, so she's talking about a short period of time. So one of the reasons why we don't end up doing this anymore is number one, we have such a difficult time of keeping things sterile. Number one, that's been our biggest problem. We are not actually gonna do it. I mean, I guess if you really want to, you're not gonna get to the end of it. So it says it's a relatively short period of time, which in reality it is, but where our plant unit comes in, we've got what, 4 weeks left of this class?
Yeah.
Essentially, for us to go through all of the stages takes about 8 weeks. So again, you're like, well, it's a short period of time. Well, we don't have enough time to do it.
Well,
Well, in reality, for being able to reproduce an entire plant,
It's gonna take
8
a hot
weeks is
minute.
pretty short. But it just doesn't fit for us at this timing. And then again, like I said, the sterility. If you really want to do it, we can demonstrate. You just are not going to see the end results of it unless you come back in the springtime.
I'll come
And then
back.
you'll be able to see it if we don't lose them all to contamination.
Should try.
Go ahead.
It's under controlled conditions, so.
What's that mean?
Well, like, I read this one thing that said like they don't use like normal like tap water, they use distilled water. They put it in like this controlled temperature with controlled light.
There you go. All of that, absolutely 100% correct.
Sterile plants cannot maintain their characteristics by sexual reproduction.
Okay, what does that mean? Sterile plants cannot maintain their characteristics by sexual reproduction.
They explained it to me yesterday, but I just forgot. But I think it means like
If sexual reproduction is happening, how many parents do you have
you have
involved?
half and half, but with this one you don't have half and half.
There you go. So you have an exact.
Rough cut.
There you go. There you go. So if we've got that one plant and we use sexual reproduction, do we have characteristics not only from the male side, but do we have characteristics from the female side coming together? So is it going to be an exact duplicate? When we do tissue culturing, it is exactly, this is the plant that I've created, and I use my poinsettias as a really good example. If you look at my poinsettias up there, they've gone through all these different breeding techniques to get the one. How do we propagate those now so we get exactly the same thing? We make clones, okay? And some of it is done through tissue culturing, some of it is done through cuttings. Huh? That's exactly what they do. They breed these that's how I've got 3 completely different varieties sitting there, is they did all these breeding techniques to get the one that they wanted, and then when they got the one that had the right look, that had the right growth ability, that all of that, then they are gonna clone that booger over and over and over again, because if you use sexual reproduction, are you gonna get traits from both the male and female part? Where if we clone it, it's just exactly like it was the previous one.
It's like a guess if you just have
Yeah.
sexual reproduction.
I'm sorry, what did you say?
It's gonna be a guess, like it's gonna be either this or it's gonna look like this.
Right, or it may not look like that at all.
Exactly.
I mean, if you think about that, you guys, I'm sure you've seen like parents, you know, a mom and a dad, and you look at the kid and you're like,
Comes back.
doesn't look like either one, because it just depends on how those genes are gonna line up and what's dominant, what's recessive in that situation. Okay, go ahead.
Rare or endangered species are multiplied by this method. Meaning, basically, they just clone the rare or endangered plant.
To make more.
Yep.
Absolutely.
Production of virus-free plants.
Now, why is that a big deal?
Because they don't have viruses.
There you go. We're not already gonna start with the illnesses to begin with, because if they're growing in that environment, do they have to be cleaned?
Yeah, they should be, unless you mess up.
You know, and again, you probably don't think about it a lot, you know, in a greenhouse, but if we think about it in livestock setting, anytime you have, and again, Shelby, this kind of plays with you with your turkeys, anytime you introduce a new set of turkeys, and you had old turkeys in there, can you bring diseases with your new turkeys and infect your
That's
old
why every
turkeys
time
with it?
we bring in new pigs from different places, we try to Jackson.
There you go. And why are we concerned with that? Because they may be, you know, and I might not show any signs of it, but I may be a carrier of it, but you've never been around that before, and then all of a sudden it hits you like a brick wall. Okay, but think about like when we go at Thanksgiving and Christmas time. What happens? We're all, you know, in school and we're doing our thing, then all of a sudden we get these breaks and we go all over the country to visit friends and family all over the place. Then we come back and we share our what? Diseases. And we're not used to it because it came from Wisconsin or Ohio or whatever, and then we all get sick and spread it around.
Oh no, I got a disease.
Oh, no. Go ahead, Katie.
Regenerates the whole planet from plants that have been genetically modified, basically saying the same thing.
There you go. So we took the time to genetically modify something and we want to make an exact clone, right?
Yeah.
Correct. Okay, go ahead, ma'am.
They're clones of each other. They're
Yep.
easy to store and transport. It's the same time to harvest, but cheaper.
There you go. So again, and that's a really good example. I got my little corn plant sitting there. I didn't need that entire corn plant. I only need a couple cells. And can I recreate millions? Pretty much indefinitely too. But that's the other thing that's awesome. It depends on the situation. If you talk about, yes, do we
Probably
have
not
to
for
have,
like Monsanto or
even
Bayer. 'Cause
even for us, Jay, to be really honest, if you think about the cost per plant,
well, we
cheap.
don't have the laboratory, do we?
no, and like I said, we've made it work in here before. It just the chances of us making it work are kind of slim. Okay, we actually did have some, and I can't remember what year it was, but we did tissue culture cauliflower, and we did sell some of our full-blown cauliflower plants out of my greenhouse in the greenhouse sale in the spring. Okay, so when you think about
That's
if
cool.
millions upon millions of plants can actually be created with one, I mean, really, you spread that cost. So we think about our turkey barns and our pig barns. Why do we have thousands of head in that barn? To spread the cost, right? I can't afford to build a barn like that and have 10 pigs in there.
Well,
I gotta spread the cost. So
Makes no
yes,
sense
this
to
is
go like
expensive, but if we can create millions of plants, that really isn't a lot cheaper than conventional. And the answer would be yes. Thank you. Are you, who's next? Jay,
Did I do
you're next.
good?
Thank you. Yes,
Oh,
ma'am.
Jay!
You should be. There you go. There's those expectations again. Katie, you have the ability, should you do it? There you go. Thank you, ma'am.
Justin, make the dog. The dog.
Huh?
The dog.
Oh, my lord.
Do it.
Tried. Remember we did that in middle school?
The dog.
Anyway, Jay Bird, what you got?
Steps of tissue culturing. So first, the scientists take a tissue or a couple cells from a plant that they are looking for, or like the one that they like the most,
So is this just like a random plant, or are we very specific about the plant we choose?
the one you want, and it'll be like a higher, really good one, usually.
Okay?
And then they use tissue put to a sterile environment and to put like a petri dish or a flask to keep it in a controlled room. Make sure everything goes right. Nothing can get out of whack, no bacteria, no nothing that can affect it.
Mm-hmm.
Next, the plant is supposed to multiply and make multiple stems and shoots desired, and then after that it makes roots, and then is able to be transferred to soil. And this picture is like they have the roots and they're starting to transfer it into soil.
So which one would be, out of your three pictures, which one would be the first one in this situation?
This one.
Right there. Okay, so when I said that they form that callus, does that little green mass of greeny yellow crap, does that look like a growing plant?
No.
Yes.
No, it just looks like this wad of cells.
Looks like grass they got clipped up and scooped in.
It looks like a wad of cells to me.
Yeah, kind of sort of, just as this wad of cells. So they took that original tissue and they put it on that nutrient media that has the sugars and the salts and all that stuff to grow, but then it's got the hormone that causes those cells to divide. They're not differentiating out into any particular. It's not like I'm making a shoot or I'm making a root or I'm making anything like that. It's just this wad of, okay? And then when they get to that point, they'll take that out, they cut it in multiple pieces, then put it in a different nutrient hormone that creates the individual plant, which is the one you see in the top. But in that one in the top, do you see any roots at this point in time?
No.
No. So then they're going to transfer it from that one. So now I have developed this. We're going to get root initiation. So we're going to transplant it and put it in there to get a new root initiation. So now all of a sudden I got my plant up here growing. Now I've got my roots. And then our last picture, we are taking those little plants that are sterile, disease-free, virus-free, exact duplicates, blah, blah, blah. It's a full-blown plant. And now I can take it and I can put it in my soil. And now I can go to my greenhouse, or I can go to my nursery, or I can go to the field, or I can go into the forest, wherever it may be that this plant needs to be placed. So a very abbreviated version. Again, this makes it look like, oh gosh, it takes 15 minutes. Okay, no,
It's
we're
a bit
talking
more
months.
difficult.
Okay, we're taking months. And again, every single time we do this, we have to maintain
Control setting.
a controlled setting in a sterile environment. Bacteria, fungus can get in there and can ruin it. And that's the thing that makes it so challenging for us, Connor. We may get the tissue cleaned and sterilized to begin with. In stage 1, we're good, we're golden, we get it through. But then we gotta transfer it from 1 to 2. And in between 1 and 2, we get contamination. Or from 2 to 3, we get contamination. So trying to maintain that, again, in this beautiful setting is a little bit challenging. Have we made it happen? The actual first time I did tissue culturing when I was down in Manning, we did not have a laminar airflow hood. We made a fake one. I used a cardboard box. We lined it with tin foil and put saran wrap around the front of it, and then sprayed alcohol to kill any bacteria, and then just did everything in this sterile little box. And we ended up with like 60-some cauliflower plants out of one little flowerette deal. And I was just like, wow, this is so easy. I couldn't believe it. It was just dumb luck. Ever since then, it's been terrible. And now we even have a laminar airflow hood. We have worse luck with the laminar airflow hood
Maybe we should just go
than we
back
did
to
with
old
it,
stuff.
with the cardboard box.
If we ever were able to get our STEM lab, then we could.
Because that bad boy should go in there and get all cleaned up and a new filter put in and whatever. It's supposed to happen over Christmas.
I know.
Is it really?
It really is. I mean, supposedly.
That's awesome.
Anyway, thank you, Jay. Who's it? Shelby? No. Yeah, Shelby's next. Oh, there's an excellent picture of what is happening in that picture, Shelby. Huh?
Isn't it brown?
It is browning from,
Yeah, so, soil.
not soil, because it's in the nutrient medium.
I'm gonna go get
What is your number 1 obstacle, Sweet Pea?
Guys being really-
There you go. Now, can we look at that and say, oh gosh, that's a fungus? Or oh gosh, that's a bacteria? Or oh gosh, that's a mold? No. But the number 1 thing is contamination, and that's what happens. You can tell that is not going well. That is not going well. You are gonna have a dead plant, okay?
No.
So, Shelby, what you got for us here, ma'am?
Contamination, well, these are obstacles.
Yes.
Contamination is the main obstacle, and then some contaminating organisms or bacteria, fungi, molds, and yeast.
Why is this such a big deal, though? I mean, we don't worry when I'm out in my greenhouse, and you've been out there. Are we all like whacked out about, oh my gosh, there's a bacteria, or oh my gosh, there's a fungus or a mold?
It's gonna ruin your whole experience.
But why?
Because it'll contaminate it and it can kill it.
You'll kill it all then?
I know, but again, why is it going to grow so well in that little environment?
Because the control is so perfect, it will grow so quickly and devastate everything.
There you go. So that nutrient auger is good to grow that little plant. But are all those nutrients, sugars, salts, and hormones also really good at growing bacteria, fungus, molds, and yeast? Yes, and if they get in there, do they just have like the perfect world to live in? They go
Okay.
bonkers. Go ahead, ma'am.
I don't really know what to.
I don't even know what that means. I don't know what it is.
It's when the plant tissue is not able to respond to the culture manipulation.
Alright, so since we can't even say the first word, can you explain to us what that means?
It is kind of when you put it in there, it's like responding to like grow. It's not growing.
It doesn't do anything, does it?
No.
No. And that does happen. Okay, just like when you guys did your transformation, did some of you get those cells to grow on that new nutrient auger? And it grew blue on the pea blue.
Well,
Yeah.
Not exactly the way
And
we wanted.
And then some of them didn't hardly grow at all, but is that the case? I mean, sometimes it works and sometimes
It's
it
called
doesn't.
trial and error.
Yeah, sometimes it works and sometimes it doesn't. Okay?
The browning of the plants, I like it, that picture.
how does it grow?
It's rotting, literally.
It's dying.
It is rotting.
Or how does it rot? There's.
Because there is a bacteria, fungus, mold, or a yeast or something that got in there that contaminated it. It is now taking over and it is just causing that plant to die and to rot. And it's.
Bye-bye.
There's mine.
And
Go ahead.
then seasonal variation, which is when the climate isn't right for the tissue culture, so like in dry seasons and when it's humid.
Okay, why would that be a huge issue if we're talking about tissue culture? Because we said it's a controlled environment in a lab. So why would a dry season or humid environment be an issue?
Cause it would be like too hot for the,
Well, is it in the lab or after we've taken the plant out of the lab and put it in the soil?
in the soil.
There you go. Okay, so in the lab's probably not gonna be a really good thing. And again, and not everybody's been in my court class, but one of the things that we are struggling, and Ms. Vine, you probably have heard Jesus talk about, if you don't harden off your plants before they leave the greenhouse, and they're sitting there in that perfect environment and everything's great, my temperature's great, my moisture's great, my humidity's great, everything's great, and then you take me outside and you put me out, it's not the same, and then what happens to me? I look terrible or I croak. So same thing, can this be a challenge? So we've made it through that whole tissue cycle, and I've got these perfect little plants ready to go, and then I take them and I put them in a not so perfect environment, and are they like hit like they hit a brick wall?
That's
Because
what
they
my
have never
thought
been
would be.
exposed to anything. Same thing when you guys get that corn in here, your corn does not look like corn. The stuff that is grown, I promise you it's corn, but it does not have the normal outside environment. So it's this really tall, skinny, spindly looking stuff. If you want it to have that look to it, do we have to have environmental impacts or environmental factors that build up its toughness? Yeah, so same thing here. This stuff is living in a perfect, sterile environment and everything's just great, and then all of a sudden, bam, you throw it in the real world, it's like, I can't handle life, can't do it, and then they grow. Okay, so again, that would be like the last part of it. We made it all through the tissue culture, we just can't survive that last part. All right,
Shelby, you did
Cutter. back. Thank you. Tanner, you're up.
Alright.
All right. So what plants do we do this with and why, sir?
Alrighty, we have a variety of different plants that we utilize this with, and there are several different reasons why we use them. And we have several different plants, as you can see listed right over here. We have everything from an oil palm to tomato. Now, for the reasons why we do this is
Breathe. Breathe.
I'm very good at reading through my
Breathe.
notes while talking. I've learned how to do it through the years. But anyhow, with such different plants, you may be thinking, well, why is there such a variety? Why do we need to grow all this? And the main reason is simply third world countries, such as Africa, where they are either refusing to accept GMOs or they really don't have the resources in a lot of areas thanks to their presidents or dictators, whatever terminology you prefer. And it's whatever
Or
you like.
governments, how's that?
Now, with such things such as a banana, they have such small seeds anymore because through adaptation and making them easier to eat for our own personal pleasure, it's hard to harvest the seeds. So what we found is that if we take the DNA, it makes it 10 times easier just to chop it off and plant it and take the DNA off it instead of taking the banana, trying to cultivate it, and then trying to plant it and everything else. And that just becomes a pain. But then it comes to the point of, why are we doing this? I understand that we have third world countries, I understand that it's hard to plant these seeds, but what's the benefit to all this? As we're talking about worker, such as on a normal farm, is estimated to be an amount of 70% cheaper to hire and to work the entire year than it is for a person that is going to be taking care of all this complicated mumbo jumbo.
Complicated mumbo jumbo.
Well, I'm being serious. It's extremely complicated, and it's not a matter of, oh yeah, we put it in a jar, we wear gloves, that's all we have to do. No, there's much, much more than that, and it honestly frightens me to some extent or another how much these people have to do. as you can see right on down here, they have all those plants growing in those jars, but to get to that point, it took so many hours of work and so much failure. And speaking about failure, we have all of our benefits and also our disbenefits with this.
Disbenefits.
Well, disadvantages, whatever terminology you really want to use in this matter of case.
Tanner's making his new and own
Well,
dictionary.
well, you got it sometimes.
Okay.
But the fact of it is, we can grow 2 times more per meter squared. Now, the reason this is such a big deal is because as we begin to look at the outlook of how much farmland we actually have available, Iowa is the #1 place for planting corn and for everything else. And it's becoming to the point of where we need to learn how to make crops that use less area instead of having a crop that will produce more. And the reason for that is because if we can figure out a way to make that use less area, we have the possibility of feeding that ginormous number right on the other side of the board here. I want to say
7.7
Yep.
billion people
7,748,830.
Million.
Yeah.
That's
748,830,108 people. And that was as of 12/6. And that number isn't going to stop growing. And I don't think we necessarily need it to stop growing. And having it slow down would be nice, but oh well. But the fact is that if we take something, we clone it so many times over, if we have such a great epidemic for plants, such as for cows having mad cow disease, we're screwed. There is no matter of, oh yeah, we'll be fine, that plant can resist it, that clone can't. But the other 1,000 of a different plant won't. So, if you take that, in a matter of fact, you're going to have a monoculture, or a single culture of plant that can't survive a certain disease. It'll wipe out the entire colony. It's not even a matter of, oh, this plant's different from that plant, and this plant's different from that plant. If we're making perfect clones, such as we're trying to do with people right now, we're going to run into a certain boundary of where we can cook ourselves to the point of where if something bad happens, or we screw up, or we have cross-contamination, whole thing's gone.
Okay, so do you really think we're gonna get to the point where we are so undiversified that we only have one crop or one plant left on the planet?
Truthfully, I believe we're going to get to the point of where we're going to find a crop that produces the most, and everybody's going to hop on the bandwagon.
Okay, but do we need different things?
We do
And
need
only
different
different plants can provide those.
Yeah, that is true.
So, again, are we probably gonna get down to, golly gee, the only plant that's gonna be on this planet is corn, and if we have an environmental condition that corn can't handle, we're all gonna die? Or is it more that maybe we have less diversity within corn, but we still have all of the other plants that we normally count on?
I don't like corn.
You don't like corn?
Corn
I
is
do,
gross.
but I'm not just.
Now, the fact that also matter comes into it is that we do have a greater failure rate on this than we do of normal planting. But the fact of the matter remains is that you can see this fail more soon or more often than that of normal planting. To where this you can see, I believe they said in the first week or two, you can see if there's going to be a seed failure or cross-contamination. To where they know, okay, we screwed up on this batch, we're just going to check the entire thing out and start over. Well, normal planting, you really don't know until it either sprouts or it doesn't. There is no real like, oh, I think something's coming up. It's a matter of if it does or doesn't. It's no in between. Which is truthfully nice about this, but it's also very difficult to have in this because you have to have a very specific facility that is tighter than NASA on constrictions about germs and everything else. And holy cow, is that a ginormous complication of a huge document. I think it was a 48-page document by a former NASA scientist that explained
Did you read it?
the decontamination process of how they're going to grow this.
Okay, so just FYI, I'm gonna throw a little light in on this. So, I made this happen in my classroom with a cardboard box, tin foil, saran wrap, and alcohol.
Did you read the 48 pages?
So,
Yeah, it was actually really interesting.
and I mean, that may be, and being sterile is a big deal, but can we, I mean, I made it happen with a cardboard box, tin foil, saran wrap, and alcohol.
Yeah.
So in reality,
Yeah, they're doing
is it, I mean, is it possible? Now, what did I tell you? Why don't we currently do this all the time in my classroom? Because we cannot maintain sterility throughout the entire thing.
Yeah.
And is that a big deal?
Yes.
But can it be done and it not be as hard as NASA going to the moon? It can be.
It can be achieved, but they are also doing it on the, I believe it's the ISS space station, and they have very tight constrictions, and it was an interesting article to read, it was actually quite fascinating.
Okay. So in a nutshell, Tanner, why do we propagate plants through tissue culture? Why do we? Not why don't we, why do we? And what plants are?
The reason we do it through tissue culture, and the main reason that we've been trying to perfect it throughout all these years, is because one, we can get a greater number per smaller area, and two, we can have bigger numbers in a smaller amount of time.
Thank you. Okay, so we're gonna take a time right there, because if you look, we're gonna run out of class time. When you think about that number, and Tanner referred back to that number, do people wanna go live in the desert?
No.
Do they wanna go live in the swamp? No. Okay, they don't. They wanna go live where the land is nice, where the climate is nice, things like that. So that narrows down the amount of space that we have to grow crops. If we can grow twice as much in a small area, can we create more environmentally controlled areas to raise crops? So we have turkey barns, we have pig barns. Could we build greenhouses that could house multiple plants in a smaller space and then provide that proper environment? So if we are in the desert and my plant ain't gonna grow in that soil in the desert, is it easier to be able to build that
Why
confined
don't we do
space in
that?
order to do that? What?
That'd be actually a good idea.
It is a great idea.
To
But
just
here's
do
the
like
thing,
giant
if we
greenhouse
have the
farms.
ability to do this, to make twice as much in a smaller area, does that make it a little bit easier?
It's efficient.
It is way more efficient.
Except for your-
So again, why do we do it? He said lots of advantage in third world countries, absolute, 100%. Okay, because if we can grow more in less and we can put up these confined, spaces or environments that are controlled, can we raise more crop for those people? Yeah. Could we do that with normal seed production like in a cornfield? No. Okay, no they can't. So there's some definite advantages to this. So our last one, and we are going to run out of time, so I guess what I would like you guys to do, on this last slide, it says, we all eat chicken nuggets. Golly gee, thank you, Connor Tagles.
No, it was not just me.
Anyway, what I would like you guys to do, because we're going to run out of time, I want you guys to pop onto this before tomorrow and add your 2 cents. What you've learned throughout all of these, I want you to add your 2 cents. Fellas, ladies, get your poop together, man. Okay? How can tissue culture be used to combat world hunger? I want each one of you to put your own response there, and then you need to make sure that your name is with it.
Yeah, we do part.
Before class tomorrow.
How many? Just one bullet?
You just have to have one bullet. What you know about what tissue culture is, the advantages of it, the steps of it, the crops that are currently being used, the obstacles that we have to overcome. In your mind, how can tissue culture be used to combat world hunger?
Efficiency.
Okay? You need to add your spot. Make sure your name is with it before class tomorrow, because we're simply going to run out of time. Tanner?
Can I use the restroom?
Yes, you may.
Alright.
Obviously, don't have time to set this up, so as soon as we get done discussing that tomorrow, we will be putting our little European cornboard eggs on our plants, and we will
Yeah,
get
just
that.
make sure it's like no of our responses like any other.
Correct, I don't want you to have the exact same thing as somebody else. So whoever gets up there first,
It's gonna have the easiest
they
time.
have the easiest time. So you procrastinators,
Yeah,
this will be more of a challenge for you. Put the light on, Connor, please. Thank you. Everything put away, turned off.
Yeah, turned off, unplugged. Well, it's still back.
Thank you.
Everything's locked.
Thank you.
Well, you were nice, thank you.
Thank you.